let me know one thing. What is your normal heart rate? 72 or? Okay, if it is 72, means in 1 minute your heart beats 72 times in 1 minute. How much it take for generation of one beat? How much time it takes? 60 divided by 72. How much it comes? Okay, now you do one thing. Suppose my heart beats at the rate of 120 beats per minute. Then how much time it will take to beat? 0.5. So initially it was how much? Okay, now your heartbeat is 75. How much it will take? 0.8 seconds. If your heart beats at the rate of 40, when your heartbeat increases, what happens to your cardiac cycle? Decreases. When your heartbeat decreases, cardiac cycle length increases. Okay. For better understanding, we'll consider our normal heart rate as 75 beats per minute. Then what would be the cardiac cycle length?
0.8 seconds.
Fine. So normal cardiac cycle length is 0.8 second. Okay? What is cardiac cycle? During every beat, some changes taking place in atria, also in ventricles, and these events are repeated in the next very heartbeat, and it goes on. These events that are taking place in the heart, in the atrium, also in the ventricle, these events together are called cardiac cycle. That are repeated in same events are repeated in every cycle. So this is cardiac cycle. So in one beat, heart is having atrium and ventricle, and they contract, relax separately, not at a time. So there are some events taking place in atria, as well as some events are taking place in the ventricle. Now, what are the events taking place in atria? Systole and diastole. And what are the events taking place in ventricle? Also systole and diastole. Okay? Now you see this circle. Suppose cardiac cycles started from here. So when this cycle will be completed, how much time will be going? 0.8 seconds. Similarly, ventricular contraction starts from here. Contraction, relaxation means diastole. It will be ending here. So both will take same time. Okay. But the events are not similar. The events are different. During every cycle, one type of change will be taking place in the heart, that is mechanical change. What change? Mechanical change. We will also see during these events, some pressure change is also taking place within the chambers. Because blood flows because of the pressure change. When there is a pressure gradient, then from higher pressure to lower pressure, it will move. And Every chamber, in its exit point, there are valves. Atria, there are exit point, there are valve, atrioventricular valves. Similarly, ventricles are having its exit point. What are these? Pulmonary artery, the semilunar valves, and aortic valve and pulmonary valve. These are semilunar valves. Okay, so they also open when there is pressure difference. When there is pressure difference, and they direct the blood in one direction, not backward. So this is the basic concept. So blood will flow from higher pressure to lower pressure and in one direction, not to the back. Okay, fine. Now let us see what are the events. Only atrium is having only two events, systole and diastole. Ventricle also having systole and diastole, but there are many subdivisions. During atrial systole, we will get three subdivisions, three phases. During ventricular systole, we will get three phases. And what are these phases? You open your diagram that I provided. First one is isovolumetric phase. See, what is this phase? Isovolume. During the early part, initial part of the ventricular contraction, early phase, there will not be any volume change within the ventricles. Means it will act as a closed chamber. The outgoing paths are closed, but it is trying to contract or eject its blood, but the valves are closed. Okay. Now see, in another diagram, I have given the ventricular blood where it will come. Left ventricular blood, it will come to aorta. Left ventricular blood, when left ventricle will be contracted, its blood will be coming to aorta. When the blood is coming to aorta, then pressure will be rising. And what will be the maximum pressure? I have given one diagram. Yeah, it is given there. Maximum pressure in aorta is 120 millimeter mercury. And when blood is not entering into the aorta, that is the minimum, that is the diastolic pressure. What is this? 80 millimeter of mercury. And now you see that what is the pressure, maximum pressure and minimum pressure the ventricle can generate within the left ventricle. What is this? It's almost like systolic pressure of aorta. How much? Left ventricular pressure. Left ventricle, see the pressure? 120. And minimum pressure is 80 or 70. That is, when blood is entering aorta, the pressure will rise to 120. When blood is not entering from ventricle into the aorta, there is blood. The pressure is 80. So during diastole of the heart, pressure within the aorta, so during diastole, the blood from aorta should come back to the ventricle, but it is not possible. Why? Because semilunar valve will close. If it tries to come back, then it will close. Semilunar closes, there will be closing of semilunar valves. Okay, now let us see, we will discuss every phase in detail. Now you go through the circle. ও যখন রিল্যাক্স করবে তখন তো প্রেসার ফল করে যাবে। ফল করে এতটাই হতে হবে কমে যাতে এট্রিয়ামের থেকে কম হয়, তবেই তো একটু ফ্লো করবে। লেফট ভেন্ট্রিকল রাইট ভেন্ট্রিকুলের মিনিমাম যখন ডায়াস্টোলে যাবে তখন মিনিমাম প্রেসার কত? খুব বেশি ডিফারেন্স না, সামান্য। লেফট ভেন্ট্রিকুল একটু বেশি থাকবে, রাইট ভেন্ট্রিকুল একটু কম থাকবে। হোয়াট ইজ দ্য প্রেসার? নাও সে this is the ventricular systole, and how much for this, how much time is taken? 0.3. And for diastole 0.8. For 0.3 second from here to here, 0.3 second, make a circle divided into 8 parts. Every part will be 0.1. Every part will be 0.1. So this is one part, this is another part, this is another part. So this part is ventricular systole. Now during this phase of systole, there are 3 more phases under this. First phase is isovolumetric phase. Means no blood will be coming out during this phase from the ventricle into the aorta or in the pulmonary artery. So this is isovolumetric phase, that's why. Why? Well, all the valves are closed, but it is contracting. It is not, left ventricle is not capable of sending its blood to the aorta. Initially, when it started contracting, its pressure was how much? 8 to 12? Now it is contracting, pressure is developing. Cannot enter in the aorta. Why? In aorta it is 80 millimeter mercury. So it has to, by contract, it is to develop force, pressure. When this pressure will be in excess of 80, then only this blood will start to propel into the aorta. So before this, what is this called? When no volume, no blood is entering to the aorta, but ventricle started contracting, it is called isovolumetric phase. It is contracting, but no volume change within the cavity of the ventricles. So that is why it is called isovolumetric phase. So when the pressure increased more than 80, means it is exceeding the pressure within the aorta, now it starts. But it is also continuing is generating its pressure. Ultimately, how much pressure will be generated? 120. So, lot of blood will be entering. Initially, flow of blood will be more rapid. That's why first phase is called rapid ejection phase. Rapid. So, initial isovolumetric phase, it is for 0.05 second. And the later part is of 0.04 second. And the next part, the last part, is the reduced ejection phase. Means here, volume is reducing, pressure is, maximum blood has entered into the aorta. So still it is trying to eject more blood. Okay, so ultimately, how much blood will be? Our ejected ventricle can contain 130 ml. How much? 130 ml. And it can send near about 70% of his blood to the aorta. How much? 70%. So what is the 70% of 130? Near about 80? Near about 80 roughly. So it is 80 ml. So in one stroke, ventricle could send how much blood to the aorta? 80. And this is called stroke volume. This is called stroke volume. Before systole, it is 130 ml. And this before systole, so there was diastole. During diastole, it has accumulated blood. Left ventricle. How much blood it has accumulated? 130 ml. So this is called end-diastolic volume. At the end of diastole, what is the volume of left ventricle? 130 ml. So this is the end-diastolic volume. So after its ejection, after completion of ejection, means after completion of contraction, how much blood will be retained? 130 minus 80 ml. So this will be the end systolic volume. In echocardiography report, you will find, the report says ejection fraction. Ejection fraction means out of 130 ml, how much you could send to the aorta. If you can send it 60 to 70 to 75%, this means your heart is strong. Your efficiency of cardiac muscle is strong. If it comes down to 40, then it is strength of cardiac muscle is reduced. It is very hard for the heart to send its blood to the aorta. Means in the periphery, there is lot of pressure, lot of resistance. Okay, so the patient or the heart may be suffering from some diseases. Okay, so how much time? Total time required from here to here? 0.3. Out of this, within this there are 3 phases. The first one is isovolumetric phase. What is the time? 0.05 second. And the next phase is rapid ejection phase. Blood will be ejected very rapidly. And what is the time? 0.14 second, 11 second. And the later part of ejection is slow. And how much time it will take? 0.14 second. Okay, so this is the time distribution all of different phases. Okay, then what will happen? Diastole will begin, and it will diastole is started from here from the end of ventricular systole. It will be continued up to this. Okay, so how much time for this? Total 0.8, 0.3 gone, 0.5. Now in this phase, during this diastole, there are sub-phases. One is protodiastolic phase. Next to this is isovolumetric relaxation phase. Then rapid filling. Now from rapid filling means from atrium blood will be coming, entering within the ventricle. Ventricle is relaxing. From where blood will come? From the atrium. So initially, valve is open, so rapidly blood will be entering from atrium into the ventricle. So initially it is rapid, so that's why it is called rapid filling phase. And thereafter, entry filling become slow. That's why this phase is called diastasis. This is called diastasis. So this phase is called diastasis, means slow filling phase. And thereafter, what happens? Last rapid filling phase. So initially, filling phase, initially there is a protodiastolic phase, no blood is entering into the ventricle is relaxing, but no blood is entering. Then what happens? Isovolumetric phase, no blood is entering again, trying to relax, no blood is entering from atrium, then valve is opened. Every valve is opened, now there will be rapid filling. Then during rapid filling and slow filling, 70%, 70 to 80% of the atrial blood will enter. And this entry doesn't require auricular contraction, it enters passively. Now you just imagine, ventricle is relaxing, there is AV valve, and these AV valves are open. So since the valves are open, blood will enter rapidly. Okay, it will enter rapidly. So initially, there will be rapid flow, and thereafter it will be slow. And again you will find, again there is rapid entry. Last rapid entry. Why this has happened? Because by this time, at the end of this time, atrium started contracting for only 0.1 second. Atrial contracting, so its blood is entering into the ventricle. As a result, there is rapid filling. There is rapid filling. So that's why this is called last rapid filling phase. When it is entering rapidly in the last phase, you will hear the fourth hour sound. And you will also hear the third hour sound, but not by stethoscope, by the phonogram. It is not audible. Third and fourth is not audible. Only first and second is audible. First one is because of closure of AV valves, and second one is because of closure of semilunar valves. And fourth one is rapid filling phase. This is the third one. Rapid when, see, Initially, ventricle was cavity is empty. Now, when the valve is opened, rapidly blood will enter, and this will cause vibration on the wall of the interventricular septum. Vibration, and because when it will enter, there will be turbulence, and this will cause vibration of the septum. Okay, so using phonogram, you can hear this. And what is this sound? Third heart sound. Again, in the last phase, some more blood will be entering because of contraction of the atrium. So again, you will be hearing another sound. This is the fourth heart sound. Okay, so let us summarize what are the different phases. It is not given for atrial phase. We know it is very simple. Point 1 is systole, atrial systole. Point 7 is atrial diastole. Okay, and ventricular events. Systole is having 3 subphases. One is isovolumetric contraction phase. Next one is rapid ejection phase, and third one is slow ejection phase. Okay, and during diastole, ventricular diastole, there are how many phases? 5 phases. The first one is protodiastolic phase. What is the time? 0.04. And what is the next phase? Isovolumetric relaxation phase. It will be relaxing, but volume will remain same. That's why it is called no blood is entering from atrium to the ventricle, because still the ventricular valve is closed. Okay, then there will be rapid filling phase, 0.1 second, and slow filling diastasis is a 0.2, and the last one is 0.1 second. Why it is 0.1? Because this blood comes from contraction of the atrium. That's why it is 0.1 second. So this is the mechanical events. We could understand what are the different phases, what are this name given, and when first heart sound is heard, second heart mechanical, this is mechanical event, sound, production of some mechanical event. Now in our next class we will study pressure changes, volume changes, and ECG changes. In ECG, in electrocardiogram, what are the different waves? Which one is S wave?
So this one.
Which wave? This is P wave. And first downward is Q. And second upward is R, third one is S, and it is T. Now during this cardiac cycle, we will see during which period P waves come, during which period R, Q, R, S comes, so during which period P waves comes. And this P wave represents auricular activity, and Q, R, S, they represent ventricular activity. Okay, ventricular depolarization, ventricular contraction. P wave indicates auricular depolarization and auricular contraction. So during this phase, it will be depolarized and it will be contracting. Okay, so after completion of this, ventricle will be depolarized, then contraction will begin from here, from the mid portion of the R wave, contraction of because it starts, contraction, mechanical process, electrical process starts early, but mechanical process influenced by electrical activity starts later, a little bit later. Okay, and T wave is because of a repolarization of the ventricular muscles. Okay. Repolarization of ventricular muscle, and this is depolarization of ventricle. This is depolarization and repolarization of atrium. So we'll see in our one graph is given. One graph is shared. What is the name of this graph? What is the name of this graph? উপরে লেখা আছে Uyghurs diagram. We will, what is there in this diagram? One is pressure curve. Pressure in atrium, pressure in ventricles, pressure in atria, aorta. So during cycle, this cardiac cycle, how the pressure changes in these cavities means ventricle, aorta, and auricles. Okay, and there is another change. There is a pressure change, volume change as well as pressure change. And what is the other thing? That is the electrical activity, the PQ, ECG changes. So we will explain this in our later class. This will be explained. When your question will come like this, you may face this question like this. What are the different ventricular events? Explain pressure and volume changes that are taking place within the ventricular cavities during these events. So you have to describe why the pressure is rising, why the pressure is falling, again it is rising, why the volume is changing. You have to explain these changes. What are the reason of the changes? Okay, so volume mainly in your syllabus it is there. In your syllabus, only they have given emphasis on two things. One is pressure change, another is volume change. So we will explain first these two, then we will change a mechanical change, another heart. When, during which period you hear the first sound? This is MCQ question. When during which period of cardiac cycle you hear the first sound, heart sound? You hear the second heart sound. During which phase you hear the fourth? Why it has happened? What are the causes of these sounds? First sound, what are the cause? Second heart sound, what is the cause? Third heart sound, what is the cause? Fourth heart sound, what is the in which phase? So you have to be very clear about this. And the pressure changes curve. So at the left side you will see in the pressure change, what are the graphs? How many graphs are there? See, one is red, one is green. Red is for atrial pressure, not atrial, aorta pressure, pressure in our aorta. And the green one is pressure in the ventricle. See, the pressure in aorta, this is what? During minimum 80. And it is rising up to 120. Again it is falling. When it is going to diastole, then it will fall. So it will rise, then fall. Rise from 80, it will fall. So you can easily remember this. When the pressure will rise, when ventricle will be ejecting blood? During ventricular systole. So you can easily explain this. And the volume and the pressure within the ventricle, we said कि minimum pressure is 8 to 12 and the maximum pressure is just identical as aortic pressure, arterial pressure. Okay, similarly, we will explain the volume change. Similarly, see, there is ECG changes. During you have to, when TU is seen, when QRST is getting, when PU is saying, you are getting. So you have to explain this. And with volume change, this one is volume change. Okay, what is the volume? During systole, diastole, one is, before the chart is given. Yeah, so you try to remember this. That's all for today. The graph will be explained in the next class. We will be performing another amphibian experiment. Based on property of nerve muscle. And this is, here we will study effect of two successive stimuli. We know stimuli are of, it may be subthreshold, it may be up to threshold, or it may be suprathreshold. If we perform an experiment with subthreshold stimuli, and if both the stimuli are given very closely very close, time gap is very minimum, so that second stimulus fall within relative refractory period or absolute refractory period. So at this time, what will happen? Both the subthreshold stimuli will combine, will be summated. As a result, now both the stimuli together will reach to the threshold limit. Now they will be able to produce the action potential, muscle contraction. If you apply the subthreshold stimulus to the muscle, what will happen? You will not find any action potential, means there will not be any muscle contraction because it is subthreshold. But I am applying two subthreshold stimuli within the latent period, within absolute refractory period. So that at the time, what will happen? Both the stimuli will combine and will exceed threshold limit. Now it will be able to, both together will be able to produce the response. Okay, so that is not our today's experiment. Our experiment is on both the stimuli are of threshold limit. It may be suprathreshold. It may be of suprathreshold. Now, before we explain this thing, what happens, what is the effect, what are the effects of these two successive stimuli? So before that, we have to learn about the action potential that is developed in muscle following stimulation. We know skeletal muscles are excitable. These are having excitability. So they are excitable cells. Means if I apply a threshold stimulus, it will produce a response. What type of response? Action potential. Now this action potential that has been developed within the muscle, so this action potential will travel along sarcolemma and they will enter into the sarcotubular system and will cause liberation of calcium ion. Now the calcium ion will bind to its active site that is present in actin, thin filaments, and when it binds with this site for calcium, then the process of contraction starts. So liberation of calcium ion is essential. For liberation of calcium ion, what is essential? Transmission of action potential. For development of action potential, what is needed? Muscle has to be, the permeability with stimulation, permeability of the sarcolemma has to be changed. In sarcolemma, means muscle membrane, there are various ion channels. One of the important ion channel is sodium channel. Sodium channel. So now the sodium channel are of different kinds. One is leaky, another is gated. Both are very important in generation of action potential. This is the nerve fiber, exolemma, exoplasm. So we have sciatic nerve. So we are putting stimulus on the sciatic nerve. So I am giving a electrical stimulus which is of suprathreshold or threshold strength. So it will definitely produce response. Now what will do? This electrical stimulus, it will cause change in permeability of the membrane. Okay. So initially leaky channel will open. As a result, what will happen? Sodium will enter within the exoplasm. So inside is negatively charged, sodium ion is entering, so negativity will be reduced, means there will be depolarization. When this depolarization, so what happened? Here stimulus is given, here I have given the stimulation, permeability is changed, sodium ion started entering within the cell and depolarization started. means it is becoming gradually less negative. And when this membrane potential coming to the threshold limit, then what will happen? So there is a threshold limit, say about 40 minus 40. When the slow depletion will arrive to the minus 40, it is the threshold potential for muscle. So what will happen? Now there will be opening of voltage gated sodium channel. So now sodium will start entering within the muscle cell rapidly. So there will be an overshoot of membrane potential, and it is reach up to 30 millivolt or plus 35 millivolt. So this is the depolarization part. Initial why it is slow? Initially it is slow. Why? Because there is no opening of voltage gated channels. Okay, there is slow, or what is this called? Leaky channels. Okay, they are only open up leaky channels. That's why these are very slow and number is less, relatively less. But when this potential comes to the level of threshold limit, then all the voltage gated channels, sodium channel open. Now there will be rapid entry of sodium ion. So this overshoot you will get. Okay. So after that, when it reaches here, sodium channel closes, voltage gated sodium channel closes, and immediately potassium channel open. So no more entry of sodium ion. Now what will happen? Exit of potassium ion. As a result, here what is happening? This is repolarization. And why this repolarization happens? Because of efflux of potassium ion. So potassium is coming out of the cell with its positive charge, so inside will be gradually negative. So there will be repolarization. Okay, and ultimately it will reach to the basal level. Even after basal level, in case of skeletal muscle, some more potassium ion enters. So now it is hyperpolarized. Now it is after hyperpolarization. So after some time it will be again coming to this level. So now what will happen? Sodium that has been entered within the cell, they are trapped. Potassium that has gone out, they are also trapped outside. So now what I have to do? We have to redistribute to the former position, to bring those ions to the former position. So now sodium potassium pump will be activated. Okay, so up here what will happen? Activation of, not only here from it starts, activation of sodium potassium pump. As a result of this, again resting membrane potential will be achieved. Now the important part is this: you have given stimulus. You have given stimulus here. The process of developing action potential started. If you apply a second stimulus before completion of the action potential, before completion of this action potential, what will happen? Whether it will take this stimulus to produce another action potential, or it is going to modify this action potential, it cannot modify this action potential. Because here also they follow all or none law. Whenever it will be stimulated, it will show this response. Same magnitude. Okay? Otherwise it will not produce any response. If it is subthreshold, it will not produce any action potential. Whenever it reaches the threshold limit, then it is same type of action. Whatever be the strength, you are given stimulus of threshold limit, same appearance, same phase, it will be there, same change up to plus 30 millivolt or 35 millivolt. Where if you apply a second stimulus, here, you see, here you are applying, here you are applying, here you are applying, they will not produce any effect. So now we call this period, this time, as a refractory period. They remains in refractory state, unresponsive to the second stimulus. So it begins. When this period begins, refractory period, when this potential reaches to the threshold limit, suppose here is the point, it reaches the threshold limit. So from this point to 1/3 of the repolarization, 1/3. So suppose this point is the 1/3 of repolarization, then this is the 1/3 of repolarization. So what will happen? This position, this period is called absolute refractory period, during which no second stimulus, if I apply during this period, I will not get any second action potential, or this action potential which is under process, this will not be modified. It will go on. It will first complete its contraction, then it is ready to take another stimulus. So this is called refractory period. I think you understood this point. Anybody is having? Otherwise I am asking you. Refractory period. When it starts? When the potential, slow potential rises to threshold limit. So from that time, and how long it will continue? Repolarization 1/3, when repolarization is up to 1/3 of repolarization, from to that period, it is the absolute period. Why we call it absolute refractory period? No second, within this period, if you apply second stimulus, this will not going to produce any effect, any response to those, because the first response to first stimulus is already going on. Already, unless it is completed, it will not take any second stimulus. Got the idea of this? Now, the relative refractory period. So from here, relative refractory period starts. So if your second stimulus is of suprathreshold value, then it can produce, relative means here it will depend on the strength of this stimulus. If the strength of the stimulus is higher than the previous one, then it may produce the response. That's why it is called relative. The thing is relative to the first one. The first one is having particular strength, and it is suprathreshold, above the threshold limit. But second stimulus, if it is more than the strength of the earlier one, this is going to produce the effect, that too during relative refractory period. Is that clear? Summation, addition. Summation means addition. Addition of what? Addition, in this experiment, there can be two types of addition. There can be two types of addition. One is addition of stimulus. Means this stimulus is of one particular strength. Second stimulus is another strength. So both of them may combine together. When they will be combining together, then they produce, since their strength is higher, so they can produce different response when they are together. So this is called summation of stimulus. Two stimuli are summated. As a result, their strength has increased. As a result, it will produce, sir, you said a action potential is developed. It will be of same magnitude and having same nature. Then if you also said, what you said? I said ki the response will be same, whether it is threshold or suprathreshold, response will be same. But you are telling now, the effect will be, when the two stimuli is added, the effect will also be added. There will be motivated. Then why? How come? My statement is contradictory. All none says, no, whether it is threshold or suprathreshold, response will be same. But now I am telling, if two stimuli of suprathreshold value are joined together, summated, then response will be more. I am contracting my bicep muscle. Easily I could do it. Okay? Here number of muscle fibers involved is less. The strength of stimulus that came from my brain is not much. Only less, there is no force there, additional force there, that's why I will be contracting few muscle fiber, contraction of few muscle fiber will do. But when I am taking this, the force is more. So with those muscles, contraction of those muscles, it is not possible to lift it. Then what I have to do? I have to involve more muscles. So second time the stimulus will be coming, it will involve more muscles. First stimuli, it caused only contraction of, मान लो, 5 muscles. When the second stimulus, which is of higher strength, now it will be adding another 5 muscles. Now total how many muscles will be combining? Who will be contracting? 10. Now force will be more. So more muscle, motor unit are actually involved there. Is that clear now? So if you give one stimuli, now this will cause contraction of a number of muscle fibers. If you give another stimuli of same strength simultaneously, now more motor unit will be involved, so more force of contraction will be generated. Is that clear now? Okay. Now we are coming into the first summation. It is clear now, summation of stimulus. Another thing is summation of effects. Summation of given stimulus and it will produce one effect. Now what will happen if I give second stimulus? What effect will be produced? It will depend when I am applying the second stimulus. When? You are applying second stimulus, but when? So this is the simple muscle curve. In a simple muscle curve, there is a point of stimulation and there is a point of contraction and there is another point of maximum height of contraction and lastly there is another part which is point of relaxation. Second stimulus, where I can apply? I say it depends. What will be the effect? It will depend on where I am applying the second stimulus. I may apply the second stimulus here after completion of effect of the first one. Okay? I may give second stimulus in here during the period of relaxation. I may give second stimulus here during contraction period. I may give second stimulation here or here within the latent period. Early part of latent period, initial part of latent period and the or later part of the latent period. Understood? What type of response I will get with the second stimulus? Because of second stimulus, it depends when I am giving the second stimulus. Whether it is after the effect of the first stimulus or during the process of the effect of first stimulus. So during the process, what is that? There is latent period, contraction period, relaxation period. So I can give the second stimulus during relaxation period. Also I can give during contraction period or I can give in the latent period, early part or in the later part. Okay. Now let's see what happens when I am giving second stimulus just immediately after the completion of the first response. First twitch, first completion of the muscle contraction. Now what happens? Here what we will find, here height of contraction is this much, this much. Here also I am applying second stimulus. As a result, here also I will be getting another contraction. Here what you will see, I will not say, you will say what is the effect. After the completion of first contraction, that is after relaxation is completed. Okay, so if I give the second stimulus immediately after the completion of the first response, first contraction, then what I will get a second curve. What the difference you will give notice between the first curve and second curve. You will see the curve and you will particularly give emphasis on height. Height of the second curve in response to the first curve, whether it has increased or it has decreased. So these are the points I am telling you what to be observed. Okay, so when I am giving the second stimulus during relaxation period, after relaxation period, what is happening to the height of the second curve? So stage 1 over. Stage 2, I planned I will give the stimulation during the relaxation period. When relaxation is going on, at that time I will be giving. So now see the response of the second contraction. Then again I am giving the stimulus during contraction period. Now see the response. And again I am giving, and in another settings, I am giving second stimulus during the later part of latent period. So latent period this much. So this is the later part, this is the initial part. So I will give stimulus to the later part. What is the response? Then I will give stimulus to the initial part of the latent period. Now see the response. In phase 2, height of this and height of the second cut. What do you think? Increased or same? This is I gave the second stimulus here after completion of the first curve, first contraction. What happened to the height? So remember this point. Why it has increased? You have to know this. You have to explain it to me. First I will explain it to you. When I applied the second stimulus, after the completion of the first contraction, that is after completion of relaxation. Here I have applied the second stimulus. It is completed. Now see the second curve I got because of the second stimulus. What is the height? Whether it is increased or decreased? Increased. So you have to know why. You have to ask me, sir, why it happened? Why it has happened? Okay. Now I am coming to this phase. I have given second stimulus in relaxation phase. Both the curve merged. One part is from initial contraction and second part is from second stimulus. Now see the height. See the height of first contraction. Less. Second contraction, more. So here also, if I give the second stimulus, even during relaxation period, the same effect I get. More response I get. Okay, now I am coming to fourth. Second stimulus is given in contraction phase. It is not ideal one. This is given to the contraction. Isn't it? It is during contraction period. Second stimulus. Now see, before relaxation, again it started. So this part is the effect of the first contraction. Here is the effect of second contraction. So here also benefit goes to the second contraction. And see the height. See the height. Here it has increased in relation to this one increased. Here also this is in relation to increased. Here also this is increased even more than this. And when I give the stimulation to the latent period, later part of the latent period, what happened? Only one curve got. And this time its height is more than this, more than this, more than all this. More than all of the curves. First, second. So here, what happened? what happened? Two type of summation has happened. One is summation of stimulus, another is summation of effect. Summation of contraction. Summation of contraction is the response, effect, and stimulus is stimulus. Summation of stimulus. In our last case, what has happened? I gave the stimulus to the latent period. Now, stimulus of first stimulus plus second stimulus, they become added. Now, first stimuli, it is supposed to contract, suppose 5 muscles. Now, second stimulus also giving response, now producing response to another 5 muscles. So, now both stimuli are added. As a result, strength of the stimulus is increased and effect is also became more. So this is summation of stimulus. Strength of the stimulus when during latent period both are joined together, and as a result effect is more effect. I have seen that why more number of muscle fibers were added. Okay. Now let us see summation of effect. So this part we have seen, and if you, another question, that phase is not there. I said ki latent period, that is first half, second half. So here I am given the stimulus to the second half. If I give the second in first stimulus, it will not produce any effect. It will not produce any effect. Why? Because first stimulus, the response is in the absolute refractory period. Second stimulus, when fall within the absolute refractory period, it will not going to produce any effect. But later part of when I gave the stimulus to the later part, it means I gave the second stimulus during a relative refractory period. That's why I got the response. Now you got the idea, clarification, relative and what? Absolute refractory period. If you apply stimulus to the absolute refractory period, there will not be any response. If you apply stimulus to the relative period, then only you can get the response. Is that clear to you? Anybody? Is that right? Okay. Now let us, this is the actual feature. This way, then it downward, then this. But that curve is not like that. It should be like this. This way, then rise. Okay. So, what is your observation? Height increased when after completion of the first, here during relaxation period, the effect of second story is more. Here, effect of second part is more. Here, second stimulus is in later part. So here also it is more. And second stimulus in the first have no response, and it is simple muscle curve. So this is in comparison to the simple muscle curve, all the other curves height is increased by the second stimulus. And why this has increased? It is because the first stimulus gave some benefit to the second stimulus, second contraction. What are the benefits it got? Okay? It's like, you know these benefits when you learn about the fatigue curve, tetanus curve. Okay? In that case, first 3-4 stimulation produced given beneficial effect. Height was increased. In first 4, this is the initial one, then the second one is the third one, is the fourth one. Gradually height increased. And after that, gradually height is decreasing. Because after completion of the fourth one, the gradually fatigue sets in. So muscle is starting to get fatigued. That's why its height is gradually decreasing, and here it is unable to come to the baseline. Means it is unable to relax, means muscle is contracting but not relaxing completely. That's why it is fatigued. Unless it is relaxed completely, how can it contract fully? If it is partially remain contracted, then there will be partial contraction, no relaxation. And because of that, fatigue, we saw there. Fatigue has appeared. Now, come to our original one. Now, the beneficial, what are the reasons? Coming to the theory. First stimulation, nerve got excited. Now this excitation coming to muscle means sarcolemma. Then it is causing secretion of or liberation of calcium ion into the sarcoplasm from sarcoplasmic reticulum, isn't it? So what happens in the first case? Some calcium ion is liberated from sarcotubular system to the sarcoplasm. Now this calcium ion, lot of calcium ion now available. This calcium ion is now binding with calcium binding site of thin filaments. Okay, then contraction is initiated. Okay, so after that, what happened during relaxation? Again, this calcium ion is coming back to the sarcotubular system. So before calcium ion could come back to its original position, what I did, I gave the second stimulus. As a result, some amount of calcium already present in the sarcoplasm. Now, because of second stimulus, more calcium ion added. So now, all this calcium ion will produce more response, more muscle contraction, more contraction process. As a result, height will be increasing. This is one thing. What? Accumulation of calcium ion in the second phase. Because after the first contraction, some calcium ion liberated, but all those calcium ion could not come back to its original position, means in sarcotubular system or sarcoplasmic reticulum. Some entered or some could not enter. So some calcium ion is available that can also initiate contraction. So beforehand it is present, and because of the second stimulation, much more calcium ion will be added. Now a lot of calcium ion is there. They will initiate, make response in a bigger way. That's why we got more contraction, more effect. This is one cause. Second cause is because of temperature. Because of temperature. First contraction, it has used some ATP, heat was evolved. Now it has increased the temperature within the cell. As a result, what will happen? Activity of the enzyme will be increased, and activity of the pathways that will liberate, that will generate ATP, those pathway, activity of those pathways will all be, rate of production will also be increased. More ATP is now available. So with this increased ability, muscle can contract. So more effect. So more increased temperature, more enzyme activity, more rate of metabolic path, more generation of ATP, more availability of ATP. Okay, so this is one. Next, with increase in temperature, viscosity decreases. Increase in temperature, viscosity decreases. Now, for muscle, viscosity is the resistance to contraction. If it is more viscous, it will produce more resistance to contract. But if it is light, becomes lighter because of temperature. Now it will be easy, because resistance is less, now easy to contract, easy to produce more effect. Is that clear? So what is the beneficial effect? When second stimulation is given, second stimulus will also produce effect, more effect. Because why? First stimulation has given some benefit to the second stimulus, second contraction. What benefit it has given? It has given some calcium ion. It has given some temperature. It has lowered some the resistance. It has lowered the resistance, viscosity. That's why it has given the benefit. So this is the benefit, the first stimulus given to the second stimulus. This is the beneficial effect. Is that clear to you?